Yes, yeah, best believe. Explore and learn new things.
Cell division is the process by which a parent cell is split into two or more daughter cells. Cell division is essential for growth, development, and repair of living organisms. And there are two main types of cell division, mitosis and meiosis. Mitosis produces two identical daughter cells that have the same number and type of chromosomes as the parent cell. Meiosis produces four genetically different daughter cells that have the number of chromosomes as the parent cell. Meiosis is involved in sexual reproduction, while mitosis is involved in asexual reproduction and somatic cell maintenance. In this lesson, you will learn about the stages, mechanism, and regulation of cell division, as well as the difference and similarities between mitosis and meiosis. You will also explore some of the applications and implications of cell division in biology, medicine, and biotechnology. First, we discuss the types of cells, and then we talk about the type of cell division, and then we see some application of cell division. So, how many types of cell do we have? We have two types of cell, somatic cell and reproductive cell. Somatic cell are the cells of the body, like the kidney, the liver, the skin cells, the roots in plants, the stem, and the leaves. And the reproductive cells are the cells are responsible for the reproduction process, like ovaries, testes in human and animals, anther in plants, and the plant ovary. And all type of cells contain a nucleus, and inside the nucleus there are bodies called chromosomes, and the chromosomes contains of two threads, like we call them chromatid, and connected by a center mass called centromere. And each chromatid structured chemically by a nucleic acid called DNA and bind with a protein. And the number of chromosomes is fixed in the number of the same species. So each species has a specific number of chromosomes. For example, all humans have 64 chromosomes. And the number of chromosomes differ from living organism, from living species to another living species. So each species has a defined number of chromosomes. And the number of chromosomes in somatic cell, the body cells, and reproductive cells differ from the number of chromosomes in gametes. So the somatic cell and reproductive cells, each of them contains a complete number of chromosomes, two sets of chromosomes. One you get from the male gametes and the other gets from female gametes. And we call the somatic cell are diploid. So they have 2n, complete number of chromosomes. But the gametes, each of them contain the half number of chromosomes present in the cell of somatic cell. So when the gametes form, they get one copy of the genetic material from the father and one copy of genetic material from the father. And so each of the gametes have half number of the chromosomes, and we call this haploid, 1n, or half genetic material. So what are the importance of chromosomes? First, they represent the genetic material of living organisms. They play a main role in cell division. By knowing the number of chromosomes, we can identify the animal and plant species, because each species has a specific number of chromosomes different from the other species. Second, how many types of cell division do we have? We have two types of cell division. Mitosis, or we call it indirect cell division, and meiosis, we call it a reduction division. So first, the mitosis division is the division that happens in somatic cell. All somatic cells divide by mitotic division, like the kidney, liver, skin, pancreas in humans and animals, and in plant, the leaves, seeds, root, and stem. We all divide by mitotic division. And in the mitotic division, the cell divide into two daughter cells. Each of them has a complete number of chromosomes. So each of the daughter cells are diploid number because they contain the same exact number of chromosomes. And notice that some cells in human cannot divide, like the neural cell, because they don't create a centrosome. And we will know that the centrosome is an important organelle in the cell because it's responsible for formation of the spindle fibers, which are necessary for the cell division, and also the red blood cells because they don't contain a nucleus. So the neural cells and the red blood cells don't divide, can divide. So what are the importance of mitosis division? First, it compensates the damaged cell. It helps in completing the asexual reproduction process in some animals. And it helps in growth of living organisms. So as you was a baby and you grow up, your cells divide, and they increase in number, so they make you grow. So how the mitosis division occurs? Before starting the mitosis division, the cell go in a phase called interphase, in which is increasing its size, making some biological activity to prepare itself for the division. So the cell in the interphase prepares for division by doing some important biological process, duplicating the amount of genetic material. And then we start by the prophase. And as you see in the prophase, the chromatin reticulum or the DNA condense, and then the chromosomes appears. And by the end of the prophase, as you see, the nucleus is disappeared, or the nuclear envelope disappeared. There are threads-like or fibers appears called spindle fibers. And then each chromosome connected to the spindle fibers by the centromere. And then the second phase is the metaphase, and as you see, all the chromosomes are arranged in cell equator. Then we go to the anaphase, in which the spindle fibers start shrinking, and by shrinking the spindle fibers, the centromere split in half, and each chromatin are separated from the other. By the end of the anaphase, the spindle fibers shrunk, and two identical groups of chromosomes, each contain single chromatid, are formed. And we go to the telophase, where the spindle fibers completely disappeared. The chromatin reticulum appears again, and the nuclear envelope appears again, and then the cell divides into two. So by the end of the telophase, the cell divides into two new cells with complete number of chromosomes. So first, we have the anaphase, in which the cell prepare for the cell division by duplicating the number of chromosomes. And And then we go to the prophase, in which the chromatin reticulum condenses, then appear in the form of a long, thin threads we call chromosomes. Also in the prophase, the nuclear envelope disappears, and the spindle fibers start to appear, and each chromosome connected to the spindle fibers from the centromere. Then the cells go or enter the phase called metaphase, where all the chromosomes arrange in cell equators. Then the anaphase, in which the centromere split, and the appear of two separate chromatid, and the spindle fibers start to shrink, pulling each chromatin to the cell pool. And then the telophase, in which reversing the change happen in the interphase, in which the chromatin start appearing in the chromatin reticulum and the appear of the nuclear envelope again. And by the end of the telophase, the cell divide into two cells. Each of them has a complete number of chromosomes. And notice that before the cell division, chromosomes appear as one chromatid, and by the end of prophase, or during the prophase, the chromosomes appear in the form of two chromatid connected by the centromere. In animal cell, the spindle fibers are formed by the centrosome, and in plant cell, the spindle fibers appears or formed by the cytoplasm. So why the change which occur in telophase are called adverse change? As we mentioned, because the change that occurs in the telophase are opposite or adverse to the change occurs in the prophase. So in the prophase, the spindle fibers appears, the chromatin reticulum change into chromosomes, and the nuclear envelope disappears. And in the telophase, the chromosomes change into chromatin reticulum, the nuclear envelope return, and the spindle fibers disappear. Second type of division is meiosis division or reduction division. And the meiosis division happens inside the reproductive cells, which are the anther and ovary in plant cell, or testis and ovary in humans or animals. And the goal of the meiosis division is formation of gametes. Each of them contain half number of reproductive material or genetic material, which is, we call them haploid number. And the goal or the reason why the cells goes or enter the meiosis division is to form the gametes, which are important for completion of sexual reproduction. And the gametes are the ova and sperm in animals, or ova and pollens in plants. And the meiotic division takes place in two stages. First, meiotic division, and second, meiotic division. And as we discussed before, the cell enter the division, it passes by an interphase to do some important biological process to prepare itself for the cell division and increase or double the number of chromosomes. And then we enter the prophase 1, where the chromatin reticulum condenses and appears in the form of a long, thin, and double-string chromosomes. But in the meiosis, the chromosomes arrange in homologous pairs, so two pairs identical, the mean of homologous. They are the same, and each pair consists of 4 chromatids. We call them tetrad. And by the end of this phase, there are an important phenomena called a crossing over phenomena occurs. And the crossing over phenomena, as we will see, the chromatid are getting close and replace parts of their inner chromatid, and that cause or make a variation in the genetic material. So the crossing over phenomena is the phenomena takes place at the end of prophase 1, in which some parts of the 2 inner chromatid of each tetrad are exchanged to produce a genetic material. And the goal or importance of crossing over, it's making a variation in genetic materials among the living organisms. So then the nucleus disappears and the spindle fibers appears and connected to the chromosomes by the centromere. And each 2 homologous chromosomes in the tetrad move away. Then the cell enters the metaphase 1, in which the chromosomes arrange in the cell equator. Then the anaphase 1, where the spindle fibers begin to shrink and each 2 homologous chromosomes move away from each other. The chromosomes separate, moving to opposite pole side without breaking or without splitting the centromere. So at this point, each pole has the same set of chromosomes. So those 2 are from the father and those 2 are from the mother. So the cell originally has 1 chromosome from the father and 1 from the mother, but here the cell has 2 sets of chromosomes, homologous chromosomes from the father in one pole and homologous chromosomes from the mother in the second pole. And so each pole contains half the number of chromosomes of the parent cell. And by the end of telophase, the spindle fibers disappear. The nucleus membrane are formed around the chromosomes. And we end up with 2 cells. Each of them has half number of chromosomes. So at the end of this phase, the cell divides into 2 cells. The nucleus of each of them contains half number of chromosomes of the parent cell. And then we enter the second meiotic division, where the cell divide by the same stages, the prophase 2, the metaphase 2, the anaphase 2, and by the end of the telophase 2, we have 4 cells. Each of them contains half number of chromosomes. So the second meiotic division aims to increase the number of produced cells from the first meiotic division, where each cell of the 2 new cells result from the first meiotic division is divided again. So why the meiotic division called reduction division? Because in the meiotic division we reduce the number of chromosomes by half, because the produced cell contains half the number of chromosomes of the parent cell.
Meiosis is a special type of cell division necessary for sexual reproduction. These are sperm-producing cells of a male crane fly, a species with only 8 chromosomes per cell. During meiosis, the cell divides twice, producing 4 haploid cells containing 1 copy of each chromosome. The first meiosis division separates pairs of homologous chromosomes, creating 2 haploid cells with only 1 set of chromosomes. The second division separates the chromatids in each chromosome. The end result is a production of 4 haploid cells, each with a single chromatid from each chromosome. When haploid sperm and egg unite during fertilization, the resulting cell is diploid, having received one chromosome of each pair from each parent.
So what are the difference between the first meiotic division and the second meiotic division? The first meiotic division, before the starting of the prophase 1, the parent cell enter the interphase, where the genetic material is duplicated, and through the anaphase 1, the centromere doesn't split, and each cell resulted from the first meiotic division are haploid, have the n number of the half number of chromosomes. But in the second meiotic division, before the cell starting prophase 2, the cell don't pass an interphase. So there is no interphase in the meiotic division 2. And through the anaphase 2, the centromere split. So in simple word, in mitosis division, the cell contain the genetic material half from the father and half from the mother. And then in the prophase 1, or in the prophase in the mitosis division, the cell duplicates the amount of genetic material. So we have 2 chromosomes from father and 2 chromosomes from the mother. Then in the metaphase, they all arrange in the cell equator. And by the end of the telophase, when the spindle fibers shrink and the centromere splits, each pool take 1 copy of each chromosome. So again, take 1 copy of the father and 1 copy of the mother, and the second pool has 1 copy of the father and 1 copy of the mother. And by the end of the telophase, we have 2 cells. Each of them contains 1 copy of each of the genetic material. So we end up by 2n as we start. But in meiosis division, so in meiosis, again, we start by a cell has all genetic materials, 1 copy from the father and 1 copy from the mother. And in the prophase 1, the cell duplicate the amount of genetic materials. So we have 2 from the mother and 2 copies of the father. And by the end of the telophase 1, when the spindle fibers shrink and the centromere doesn't split, we end up with 2 cells. Each of them has 2 copies but identical. So 2 copies of the father genetic materials and 2 copies of the mother genetic materials. So we say that this has N or haploid, and this cell is haploid. So when each of the produced cell enter the meiosis 2, and in the telophase 2, when the spindle fibers shrink and centromere split, we have 4 cells. Each of them receive 1 copy of the half number of the chromosomes. So this has 1 copy of the father materials, 1 copy of mother or father materials, 1 copy of mother materials, and 1 copy of mother materials. And that's why we call the meiosis division a reduction division, because we started by a cell has a complete set of genetic materials and end up with 4 cells. Each of them has 1 copy of one of the parent genetic material. So what are the applications of cell division? First, some somatic cells, as liver cell, are not divided in normal condition, but in certain condition, they have the ability to divide. So, if the liver gets injured or a part of it was cut, the remaining cell undergo mini mitotic division to compensate the missing part, because we said that the goal of the mitotic division is compensating the damaged cell. And as scientists or in medicine, we use this ability to make or to do a liver transplantation. So when someone has a damaged cell, we take part or a portion of the healthy cell of the donor and transplant it to the injured person. And by the time the part of the cell that was transplanted in the unhealthy person move or undergoes many mitotic division to grow a liver. So a liver transplantation process is performed to replace the infected patient's liver with a part of a healthy donor's liver. As time passes, the liver of each of them completed due to the occurrence of mitotic division. We know about cancer, and the cancer, we say it's a crazy cell. So it's the cell that lost control over itself in division, so it's divided continuously, forming a tumor. So the tumor is the mass of cell produced due to the abnormal continuous division of cells. And we all know that Dr. Mustafa Said, which discovered a way of detecting and killing the cancer cell or tumors by the gold molecules in a technology called nanotechnology. So how it's done? First, the discovering of cancer disease. The scientist starts by loading a protein that can attach to the cancerous cell proteins. So the cancer cell produce a certain proteins, and we load some proteins that can attach to the secretion of the cancer cell. And then load those proteins with the nanomolecules of gold. So first, the discovering of a cancer disease. We're preparing a proteins that only can attach to the cancerous cell secretions and load those proteins with particles of gold. And when we inject those proteins to the cancerous person bloodstream, they only search the body and only attach to the cancer cells. And with the microscope, we can search for those gold particles and locate the cancer. And then the treatment of cancer, it has two ways. First, by using a loaded protein with nanomolecules with gold. So the proteins which has the gold particles attach themselves to the cancerous cell, and we can load them or we can focusing a laser to those proteins. The gold is a metallic, so it has heat conductivity, so it absorbs the heat and get heated, so it burn the cancer cell. Or by using a microscopic pump, again using a nanotechnology, we developed a smart microscopic pump that penetrate the cancer cell and explode inside them. So the mice was treated by the microscopic pump. They increase their life by 300 days. And the rats or mice that didn't receive this treatment only live 43 days.